So thanks all for coming and listening, and thanks for the introduction. I'm Íñigo, and I'm going to present my work, which is entitled Therapeutic Impact and Immunomodulation by MYC Inhibition in KRAS-driven non-small cell lung cancer with diverse mutational landscapes. So as you may know, lung cancer is the second most common cancer type, both in males and females, and is the leading cause of cancer death worldwide in both sexes. Inside lung cancer, non-small cell lung cancer represents 85% of the cases, with KRAS as the leading mutated oncogene in 30% of them. Inside the KRAS mutant tumors, there are other genes whose frequency of mutations is also noteworthy, which are the tumor suppressor genes p53, LKB1, also called STK11, and KEAP1. However, KRAS is not the unique gene to take into account in this type of cancers. MYC is normally amplified or deregulated in most cancer types, and non-small cell lung cancer is not an exception. Moreover, it is downstream of the pathway of KRAS, making it a target in these specific tumors. Mutations in all these genes alters the immune microenvironment of the tumor. So, for example, when p53 is lost, there is a higher immune cell infiltration that is the typical of the hot tumors. In the case of mutations of STK11 and KEAP1, are more associated to an excluded or cold tumors. In the case of the MYC amplified of the tumors, they are more associated also to this last group, since MYC orchestrates immune evasion. So being MYC essential in this immune evasion, all these immune features could be reverted by MYC inhibition, making it a promising strategy for treating KRAS mutant tumors with different mutational profiles and to prevent or even revert immunotherapeutic resistance. But how can we achieve this milestone? In order to inhibit MYC, my lab designed the first direct MYC inhibitor called HOMO-MYC, but how does HOMO-MYC work? This mini protein is based on the VHLH leucine zipper of MYC, but with 4 amino acid mutations that alters its dimerization specificity. The mechanism of action of HOMO-MYC consists on displacing the oncogenic MYC-MAX dimers from the DNA binding region, and this can be achieved by HOMO-MYC occupying these regions both as homodimer or heterodimer with MAX, or binding to MYC and sequestering it from the DNA. As neither HOMO-MYC nor MAX have the transactivation domain of MYC, all these three mechanisms block the expression of the MYC-related genes. So first, before assessing the effect of HOMO-MYC, we have to study the role of the concomitant mutations. In order to do that, our collaborator Silvestre Vicent from CIMA kindly provided us with a KRAS mutant cell line derived from a non-small cell lung cancer mouse model and modified by the CRISPR-Cas9 technology to obtain the knockout of the different tumor suppressor genes of interest. So in this way, we are able to work with 4 isogenic cell lines that only differ in one specific gene. So first, after confirming the knockout of the different tumor suppressor genes, we studied the growth rate in vitro of these cells. And we can see that after 5 days, they had a similar growth in vitro. Moreover, we studied the MYC levels, and we saw that the cells with the concomitant mutations have a higher increased levels of MYC compared to the parental one. Moreover, when we inoculate these cells in syngeneic mice, we saw that the KEAP1 mutant tumors presented faster in vivo growth compared to the rest of the tumors. Once we have the characterization of these cell lines, we treated them with HOMO-MYC to study the effect of MYC inhibition on cell growth. So first, we can see that all of them has a similar IC50 value, except the STK11 that presents a slightly higher one. And using 20 micromolar as a standard dose, the HOMO-MYC treatment during 5 days causes a significant reduction of the cell growth in all the KLA cell lines. Interestingly, although there was a similar therapeutic impact, the p53 mutant cell line presented less intracellular HOMO-MYC compared to the rest of the cell lines, both at 3 and 5 days of the treatment, which suggests poorer capacity of penetration of the mini-protein, but also higher therapeutic efficiency of this cell line. We later analyzed by the qPCR genes related to immune evasion, such as CD47, PD-L1, and IRF7, and also antigen presentation-related genes such as TAP1, TAP2, and ERAP1. We saw that the treatment of HOMO-MYC during 3 days did not trigger a significant change of these genes in the parental cell line. However, when the treatment with HOMO-MYC in the cells with concomitant mutations provoked a more profound alteration of these genes in all the cells with the concomitant mutations, and these alterations were different depending on the specific mutational landscape. After that, we inoculate these cells in immunocompetent syngeneic mice, and we study the effect of MYC inhibition and to see if the immune system reacts differently on each mutational landscape upon the treatment with OMOMYC. So as you can see here, mice that were treated with OMOMYC, they present a significant reduction of tumor growth in the parental, the p53 and the KEAP1 mutant cell line, and studying the area under the curve, we can see that the p53 mutant cell line has a more profound inhibition of cell growth compared to the rest of the models. In this graph, what you can see is all the studied immune subpopulations and the difference of expression in the treated versus the not treated conditions of each cell type. Besides, the size of the circle is representative of the p-value. So what we can see here is that the effect of OMOMYC, it produced a significant modulation of the immune microenvironment in different immune subpopulations, highlighting the increased expression of OX40 marker in both cytotoxic and non-regulatory helper T cells, as well as the downmodulation of the M2 macrophages. Looking in this case at the p53 mutant model, we can see that the induction of the immune response derived by OMOMYC is completely different compared to the parental model. In this case, increasing the expression of the PD-1 positive and TIM3 positive helper T cells, also the increase of the NK T cells, and in this case, a decrease of the regulatory T cells. Looking at the other two models, in which we saw a milder reduction of tumor growth compared to the other models, we did not see a significant change of any subpopulations, meaning that an enhanced immune modulation was observed in those models in which OMOMYC was able to reduce tumor growth more efficiently. As we have seen that the effect of MYC inhibition on remodeling the tumor immune microenvironment varied depending on the specific concomitant mutations, these same mutations is the factor that determines the immunotherapy to select for combination strategies with MYC inhibition therapy. So for example, in the non-targeted model, the OMOMYC-derived anti-tumor immune response was mainly generated by the upregulation of OX40. The rationale is to combine OMOMYC with an anti-OX40 agonist that could activate, that could boost this activation pathway. So as you can see by the relative tumor volume, the combination group or the treatment with OMOMYC delayed the relapse of the tumors after the treatment with the OX40 agonist, which is confirmed by the increase of the median survival up to 89 days compared to the 61 days of the immunotherapy alone. Besides, if we take the best response of each mouse, we can see that 90% of the animals that were treated with the combination saw partial or complete response against 54% of the animals treated only with the immunotherapy. Changing gears to the p53 mutant model, in this case we saw an increase of the PD-1 positive, TIM3 positive helper T cells, and also a downmodulation of the regulatory T cells. Some authors have claimed that high FOXP3 expression has a negative value for the PD-1 inhibition therapy in non-small cell lung cancer. So with this in mind, the rationale of combining OMOMYC with an anti-PD-1 antibody could trigger synergistic effects in this mutational context. So just by looking at the relative tumor volume, we can see a decrease or a reduction of tumor growth in the combination group compared to the immunotherapy, which is confirmed also by the significant increase of the overall survival of the combination compared to the immunotherapy. For example, at day 36, we had a 78% of overall survival compared to the 22% of the immunotherapy alone. In addition to this, if we take also the best response of each mouse, we can see that all of them that were treated with the combination show partial response as their best response against half of the animals treated with the immunotherapy alone, confirming that OMOMYC enhanced significantly the therapeutic efficacy of anti-PD-1 treatment in a KRAS and p53 mutant context. So with this in mind, we can conclude that the presence of these concomitant mutations increases MYC levels in the context of KRAS mutation, that all of the KRAS mutant cell lines respond to a similar extent to MYC inhibition in vitro, regardless of their mutational landscape. Also, OMOMYC alters the immune evasion and antigen presentation genes more profoundly in those cells with the concomitant mutations. In syngeneic models in vivo, we can see that the effect of MYC inhibition depending on the specific mutation that they present, and also the effect of MYC inhibition on remodeling the tumor immune microenvironment vary depending on the specific mutational landscape. Finally, OMOMIC enhanced the therapeutic efficacy of different immunotherapies, both in the parental and in the p53 mutant model, making OMOMIC a very promising partner for overcoming immunotherapeutic resistance. So I want to thank first Laura and Silvia for my two directors of this project, and also all the people from the BioLab and from the Peptomic team that have helped me develop all these results, as well as to the collaborators in CIMA, and thanks on for listening.
Is there any questions? So thanks for this nice and interesting talk. Let's start for the last one. Is OMOMIC undergoing initial clinical trials, as far as I know?
Yeah.
So have you checked whether those changes in immune contexture that you define upon OMOMIC treatment in your mouse model do translate into the patients? Do you see the same changes in patients?
So thanks for the question. Yeah, OMOMIC is now in phase 1B of the clinical trial, and with the patients that we have now at only the phase 1 that we have done, we were able to study in some of them some of the immune markers that we have here, and more of them. And it's true that we have seen some modulations of more or less the genes that we have, that I have presented here, and other genes that we have seen in other models in the preclinical data. So it's true that more or less it's going in the same direction. So that is really good to have these results in patients. Is really good.
So one curiosity is that the effect of OMOMIC is not related to the levels of MYC expression in your models. So those cell lines with higher MYC expression are not the ones that respond, are not the models that show best responses to MYC inhibition. Yeah. So could it be that whatever you're observing are side effects of the alternative mutation? Have you characterized the immune contexture in the absence of OMOMIC, and whether it does compare to the effect you see along treatment?
So thanks for the question, because it's a question that all the people ask. And it's true that at the end, if you think about OMOMIC, that is a MYC inhibitor, you could see, or you could think that MYC is the potential biomarker of this treatment, and we have seen here and in other many models that we have in the lab that it is not. So at the end, the levels of MYC is not a biomarker or anything that you could think of it is going to work better or it's going to work worse depending on MYC high or MYC low levels. So that is also a bad thing, because you will think, okay, so MYC is the biomarker, and okay. But it's not the case. But it's true that, for example, here I'm not showing this data because I think it was too much, but we have studied the immune cell compartment of all this mutational landscape without the presence of OMOMIC, just by knowing the basal levels of these immune cell compartments, and with that we were able to see, okay, how OMOMIC is changing these immune cell levels when we are treating them with OMOMIC.
And the final one, MYC is also used, so important for T cell and in lymphocyte proliferation. How does it affect OMOMIC on the immunity, anti-tumor immune responses?
So we know that at the end, for example, OMOMIC, it doesn't have any marker or any gene that could trigger specifically to the tumor. So it's true that it may affect other cells that are present in the mouse or in the patient, but we have seen both in the preclinical model and, for example, in the patients, that it is not affecting other immune cells. Also because at the end, OMOMIC has the ability of penetrating more in the tumor cells because the tumor cells have this macropinocytosis increase, so at the end OMOMIC is entered by that. So it's true that it enters more in the tumor cells than in the other type of cells, and we have seen that it has not effects in other cells. That is an obvious an obvious question because at the end it doesn't have a marker or something for the tumor cells. Thank you.
Thank you.
Wait a minute, wait a minute.
Estoy.
Don't leave. Don't leave me without my question. It's simple. The certain genes that are related with class 1 presentation are affected. So have you studied how are affected the immunopeptidome of those cells when you treat with your homomix?
With homomix. No, so by now we have only studied the qPCR because it's what we were able to do, but I think it's a good thing to study because at the end, well, you have seen that this immune modulation was different depending on the mutational profile, and it's a thing that we want to do because at the end it's something that results have led us to do all this of the preclinical data and all the in vivo experiment, so it's something that we want to do.
And class 1 is affected? I mean, are increased or decreased?
We have not seen many modulation of the class 1, so that is only the presentation of the genes that were most modulated or upregulated and down-modulated. So it's just what we've found modulated in the qPCR, but we have not seen any of the class 1 altered in these models.
Thank you. So